All right, the last biomolecule that we're going to discuss today is going to be nucleic acids. Nucleic acids are polymers that are made up of individual nucleotides. The nucleotide is going to be our monomer. So the polymer will be the nucleic acid. The individual monomer will be called a nucleotide. Shown over here is going to be a sample of DNA. DNA is just one type of nucleic acid. It's going to store information for replication and protein synthesis. And we're going to talk about some of the different forms of nucleic acids. Over here, like I said before, is deoxyribonucleic acid, otherwise known as DNA. Each cell in your body, or really in any organism, has to store information for how that cell should behave, how it should respond to the environment, how it should replicate. This information is all going to be stored in a form known of, form of nucleic acids known as DNA. So this is going to be responsible for the genetic material that stores information. RNA is also a form of genetic material, but its function is going to be a little bit different. RNA's primary function is going to be protein synthesis and the regulation of gene expression. And here's what that means. DNA is kind of like your textbook. It's got the hard copy of all the information you could ever possibly want to know about Bio 101 in my course. It's a lot of information, and hopefully when you are sitting down and you're learning a bit of information, or you say, hey, this week we're going to learn about chapter 1, I'm hoping you don't sit down and actually read the entire textbook cover to cover. There's a lot of material that's unnecessary for your purposes of learning the material in chapter 1. You're just going to focus on the little bit that you need to accomplish whatever that task is in front of you. Perhaps that task is the chapter 1 homework. RNA is going to be just the individual chapter that you need. You're going to sit down, you're going to take your notes, then you're going to apply those notes by doing your homework assignment. That's RNA. RNA is going to be the shorthand version of the same information that's in DNA, but it's just going to be a shorthand, quick notes version that's going to allow you to do whatever it is you need to do. Usually it's going to be, I need to make a protein because I need to do X, Y, and Z. Maybe there's too much glucose in the blood, and real quick I need to get it out of there. I can do that by issuing the insulin, or we can do that by issuing the hormone insulin. Well, insulin is a protein. If we need to make it very quickly, we don't want to sit down and copy out the entire textbook of DNA. We don't want to make every single protein that the body knows how to make. We just want to make insulin for our immediate purposes. And we're going to do that by using RNA. And we'll get into DNA and RNA quite a bit in some later chapters. So I just kind of want to introduce you to what their purposes are in this first chapter. Coenzymes are another really great example of a nucleic acid, another interesting purpose of a coenzyme. Their job is going to be to help regulate enzymatic reactions. So they're not actually responsible for storing information, they're not responsible for processing information, they are just going to help control enzyme related reactions. And then finally, the very last form of nucleic acids that we're going to talk about in this chapter is going to be ATP. ATP is pretty interesting. ATP has actually nothing to do with the storage of information at all. Its job is not to store information. Its job is to store energy. Weirdly enough. So I don't know if you knew that or not, but you do have an energy storage molecule in your body known as a nucleic acid, ATP. And we're going to spend a little bit of time going over ATP at the end of this chapter. But first, we're going to start off by going over the structure of a nucleotide. Now, this table is pretty fantastic. We're not going to dive in too deep between the differences between DNA and RNA, at least not in this chapter. However, this will be relevant information a little bit later on. So maybe dog ear this table in your notes and come back to this when we start talking about DNA and RNA. But table 3.4 will help go over some of the differences and similarities between DNA and RNA. Instead, what I would like to focus on for this chapter, just because we are going to come back to DNA and RNA, I want to focus a little bit more on ATP. ATP is a fascinating molecule. It's going to be composed of an adenine, a ribose, and 3 phosphate groups. So that's where we get the term adenosine. It's from the adenine and ribose, and then we have 3 phosphate groups, the adenosine triphosphate. And the interesting thing about ATP is that it's used for storing energy, and the way that it does this is because our molecule of Our molecule of ATP has these 3 phosphate groups at the end, and these 3 phosphate groups, they don't really get along. They're actually highly, highly unstable when they're all strung together like that in a row. And so what happens when we have a very unstable bond, typically an unstable bond means a very high energy bond. It requires a lot of energy to hold that in place. And when we break off that final phosphate group, a huge burst of energy is released. So this is how we can use ATP as an energy storage molecule. We can hold a little bit of energy in that final bond, and when we need energy, we snap that final bond and we release a bunch of energy, which can then be used in the cell for metabolic reactions. And we're going to use this a lot, and we're going to see this a lot. But ATP is very, very short-term energy storage. I know we said that like carbohydrates are short-term energy storage and lipids are long-term energy storage. I want you to think of ATP as like instantaneous energy storage. I don't know about any of you out there, but I think of ATP as cash. I do not have cash. I'm not good with cash. When I got my very first job, I was given a debit card and a bank account. So for me, a credit card, debit card, that is hard-earned money. I do not spend it if I don't have to. That is hard-earned money. Cash, on the other hand, burns a hole in my pocket. Cash is birthday money. Cash is fun spending money. And so if I ever have cash, it's gone in an instant. It's used for making purchases, and it's there, it's gone. If I withdraw cash, I use it. I only withdraw cash right before I need to spend it because I know I can't hold on to it. This is what ATP is. ATP is too unstable to actually store energy for very long. But it's very good at instantaneously storing energy. If we need to capture energy from the sun and turn it into a form that we can use later on, ATP is going to be the stepping point. ATP is how we turn solar energy into long-term carbohydrate chemical energy. ATP is going to be our temporary stopping point. So yes, ATP is used for energy storage, but I really want you to think of it as like instantaneous energy conversion. It's how we get from glucose into whatever it is we need energy instantly for. So it is energy storage, yes, but not long term, not even as long as, carbohydrates. That's short-term energy storage. ATP is more quick energy conversion. So we covered a lot in this chapter. It's why this last section was really kind of short. If we're going to go over DNA and RNA in later chapters, I really didn't want to spend too much time on it now because this chapter is so huge and we covered so much. So I would like you to just review, take this time to review. But in section 1, we talked about organic chemistry and how that is the chemistry of carbon-containing molecules. There are some unique properties of carbon. One, it can form up to 4 covalent bonds with other atoms, which is awesome. Specifically, it likes to form those covalent bonds with carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, those 6 most common elements found in all living things. And it also, all the variation or the variety comes from those functional groups, as we mentioned, that can change the shape and we change the structure. Isomers are organic molecules with identical molecular formulas but different structures. And if we change the structure, we're going to change the function and change the reactivity of those molecules. And then finally, we spent quite a bit of time on the synthesis and degradation of biomolecules. We looked at those dehydration and hydrolysis reactions. Please review those and be comfortable recognizing and identifying those. Here is those synthesis and degradation reactions. Just as a reminder, we use a dehydration reaction to create a polymer from monomers. So we're going to cut off this hydroxyl group, cut off this hydrogen, and we're going to spit off a water molecule. This is what's known as a dehydration reaction. It's a synthesis we're building up. In order to go in the opposite direction and break a large polymer down into its monomer components, we're going to use a hydrolysis hydrolysis reaction. It says hydration reaction. It should say hydrolysis. Water lysis, water to split apart. We're using water to lysis or split that bond apart. So we add that water, it's going to split apart into a hydroxyl group over here, a hydrogen group over here, and break that polymer down into monomers. Then we talked about the different biomolecule categories. We started off with carbohydrates. We talked about monosaccharides, disaccharides, and polysaccharides. We talked about their various functions for energy support and energy storage and structural support. Then we went into lipids. In lipids, we talked about triglycerides, and we talked about the significance of saturated versus unsaturated fatty acids. Then we went into phospholipids and their composition, the phosphate heads, the two fatty acid tails, and we talked specifically about the polarity and structure. Please review that if you are uncomfortable or not familiar with that at all. Please go back over the polar phosphate head and the nonpolar tails and how that contributes to the overall plasma membrane bilipid layer. Then we went on to talk about steroids and waxes as well. Next, we went into proteins. In proteins, we talked about how proteins are going to be polymers made up of individual amino acids. A peptide is going to be two or more amino acids joined together by peptide bonds, and that the difference in proteins, the difference in the functions of proteins comes from those functional groups that are attached to that singular chain. The structure of a protein, don't forget to go over that, the primary, secondary, tertiary, and quaternary structure. We did talk a little bit about protein denaturation. Mostly it's going to come up again when we get to our enzyme lab. It's going to come up again when we talk about enzymes later on, so just make sure you are comfortable with that term, denaturation. Again, it probably won't be on any homework quizzes or tests for this week, but you will see that again, so make a mental note of denaturation. Last, we talked about nucleic acids. We talked about their structure, how they're polymers of nucleotides. They're made up of a pentose, phosphate, and a nitrogen-containing base, and that their function is going to be primarily for energy storage, protein synthesis, and energy storage in the form of ATP. And that was it for this chapter. Please let me know if you have any questions, comments, or concerns. I'll be happy to discuss anything in our discussion meetings.